looked in the mirror and wondered, okay, how did I get my mom's eyes but my dad's smile? It's a question we've all asked, right? For literally centuries, we've been fascinated by heredity, how traits get passed down. And while it seems simple on the surface, the actual rules behind it, well, that's a whole fascinating puzzle. For the longest time, the most popular idea was something called the blending hypothesis. And, it makes sense, right? The theory was that your parents' traits just mix together, kind of like red and white paint blending to make pink. But here's the thing. That idea had a huge flaw. It couldn't explain how a trait, like your grandpa's red hair, could skip your dad's generation and then show up in you. The other idea was that traits were more like separate little particles. Think mixing red and white marbles in a jar. They're in the same jar, but they don't actually blend. So, that's our big question. Are we just a permanent, blended cocktail of our parents' features? Or is there something else, something more complex going on? Turns out, the answer came from a place you'd probably never expect. Okay, let's meet the guy who started to crack this code, Gregor Mendel. He was a monk working in a simple abbey garden back in the 1860s, but he had a background in math and, wow, a ton of patience. And with those pea plants, he became the detective who found the first real clues to solving this ancient puzzle. So Mendel argued that parents don't pass on blended traits, they pass on these separate, distinct, heritable factors. Today, we know them by a different name, alleles. Just think of them as different versions of the same gene. So for the gene that controls flower color, there's a purple version, that's one allele, and there's a white version, which is another allele. And this led him to a really crucial distinction, one you've got to get to understand the rest of this. There's the genotype, which is the actual genetic code. For example, a plant could have 2 copies of the purple allele, we'd write that as big P, or 2 white ones, little p, little p. When the alleles are the same, we call that homozygous. But what if it has one of each, big P, little p? That's called heterozygous. Then there's the phenotype. That's just the physical trait you can actually see, like are the flowers purple or are they white? So how did Mendel predict all this with such crazy accuracy? He used this simple but honestly brilliant tool called the Punnett square. It's really just a grid that lets us see the probability of what the offspring will look like. First, you just figure out the genotypes of the parents, then you determine the possible alleles each parent can pass down. You draw your little 2x2 grid, fill in the boxes with all the possible combinations, and then you just count them up. And when Mendel did this, something amazing happened. He crossed a purebred purple flower with a purebred white one, and their kids, all purple. But then when he crossed those kids with each other, he consistently got this exact 3-to-1 ratio of purple to white flowers. And this was the bombshell. The white trait didn't blend away or disappear forever. Nope, it was just hiding, masked by that dominant purple allele. This was the evidence that pretty much blew that old blending theory right out of the water. So, we've got it all figured out then. Seems pretty simple. A dominant allele shows up, it wins. End of story, right? Well, as it turns out, nature always has a few more curveballs to throw at us. Let's move on to a brand new puzzle. This time, we're going to look at the feather color of Andalusian chickens, because this case seems to completely break the rules we just worked so hard to figure out. Alright, so let's use our new Mendel knowledge. If we cross a purebred black chicken with a purebred white chicken, what do you think we're going to get? Based on what we saw with the peas, you'd probably expect them all to be black, right? They're gray? Wait, what? Gray? That looks exactly like the paint blending idea we just decided was wrong. So what in the world is happening here? Did we get it all wrong? Well, no. This just brings us to a new, really cool concept: incomplete dominance. In this inheritance pattern, neither allele is completely the boss. So when an organism is heterozygous, remember, that means it has one of each allele, the phenotype you see is actually an intermediate blend of the two. And check it out! Our trusty Punnett square is still the perfect tool for the job. Here we're crossing 2 of those gray heterozygous chickens. And you can see, the way we set up the grid is exactly the same as what we did with Mendel's peas. The mechanics are identical. And the results are just brilliant. You get 25%, a quarter of them being black. Another 25% are white. But look at that, a full 50%, half of the offspring are gray. So instead of that 3-to-1 ratio of phenotypes we saw with the peas, we get this beautiful 1 to 2-to-1 ratio. Why? Because that heterozygous BB genotype makes its own unique intermediate phenotype. So let's just put these two side by side to really drive it home. With the peas, that's complete dominance. The heterozygous PP looks exactly the same as the homozygous PP. They're both purple. But with the chickens and their incomplete dominance, that heterozygous BB has its very own look, gray. It's completely different from both the black BB and the white BB. Same tool, just a different rulebook.
So, we've covered complete dominance and incomplete dominance. But as you've probably guessed by now, we're really just scratching the surface. These foundational rules are what open the door to the much larger and way more fascinating world of genetics. I mean, there's also something called codominance, where both alleles show up fully and separately. Think human AB blood type. You've got gene linkage, where traits on the same chromosome tend to get inherited as a package deal. Then there's epistasis, where one gene can literally just turn another gene off. And get this, sometimes a single gene can influence a whole bunch of different traits. That's called pleiotropy, and it's a huge factor in complex disorders.
And that really brings us to the final thought here. We've just spent all this time untangling the complexities hidden inside a single gene for flower color or feather color. Now take a second and think about human traits, like your height or your risk for heart disease. Those aren't controlled by one gene. They're influenced by hundreds, maybe even thousands of genes, all interacting with each other and with our environment. The Punnett square, it gives us the basic language, but the real story of inheritance, it's an epic novel. And learning its grammar is the key to cracking the code of life itself.